Good morning, everyone, and welcome to this session. Let me briefly introduce myself. I am Simonetta Di Simone, a PhD student at the Department of Pharmacy in the Medicinal Plant Unit from Naturale University in Chieti, Italy. So the topic of this session is the determination of medicinal plant extracts biocompatibility through eco-toxicological assays. So let me give you an overview of a typical multidirectional study we conduct when we decide to investigate a new plant extract. So the first step is optimization of extraction, followed by the determination of the quality and the quantity of bioactive compounds, and an investigation of intrinsic scavenging and enzyme inhibition properties. All this before proceeding with the intended biological testing. So the biological investigation consists in preliminary eco-toxicological studies, followed by in vitro assays on different cell lines under different conditions, and ex vivo studies on tissues. So in this lesson, I would like to concentrate on the three different preliminary eco-toxicological studies that we conduct in our labs. But first of all, what is the definition of eco-toxicology? Ecotoxicology is the study of how environmental pollution affects ecological entities as populations, communities, or ecosystems. And ecotoxicological tests are test systems that expose biological components as cells or individuals to chemicals to register biological effects. These tests measure these responses at different levels. They can measure the response at a suborganismal level, at a whole organismal level, at a population level, or a community level. The ecotoxicological assays that we perform belong to the whole organismal level. So the first of the three assays is the allelopathy assay. That is performed in order to investigate the phytotoxicity of the plant extract under study. The second one is the brine shrimp lethality assay, BSLA, that permits us to predict the biocompatibility and toxicity limits of the extract. The third one is the Daphnia magna heartbeat rate assay, which helps us to investigate the potential acute cardiotoxicity of the extract. So let's begin with the allelopathy model. First of all, it's important to know what we mean by the term allelopathy. Allelopathy is the chemical inhibition of one plant by another. Due to the release into the environment of substances acting as germination or growth inhibitors. And these substances are called allelochemicals. Plants can release allelochemicals into the environment through different ways, so through exudation, leaching, exhalation, even by the decomposition of roots, stems, leaves. And for example, simple phenolic acids are the most common and important class of allelochemicals present in ecosystems. And in soil, these compounds may exert allelopathic effects on neighboring plants. So seed germination is the resumption of growth by the embryo within the seed that leads to the development of a new plant. Germination begins with water uptake by the dry seed and finishes with the elongation of the embryonic axis. Phytotoxicity tests can be performed using either seed germination or root elongation as test endpoints. So the two germination parameters we analyze in an allelopathy assay are the seed germination expressed as germination percentage and the seedling growth expressed as relative growth percentage. That is the mean of seedlings length compared to a control sample. So an inhibition of germination or a slowdown of seedling elongation is considered or are considered as first evident signs of phytotoxicity. But how does the allelopathy assay work? So in an allelopathy assay, seeds of plants species are placed in contact with different concentrations of extract. After a specific time, usually 3 or 4 days, this depends on the species we selected, under controlled conditions, the number of germinated seeds is counted, and germination is expressed as one or more indices. But let's look at the assay in a bit more detail. So the experimental setup. The assay is conducted in Petri dishes covered with a double-layered filter paper disc. And this disc is then previously soaked in the plant extract at a scalar concentration. And distilled water is used as a negative control. So we divide the paper disc, as you can see in the image, in 3 areas. And we put 10 seeds for each variety in each area. After that, we seal the Petri dish with parafilm to ensure a closed system model. And we incubate them in darkness at room temperature for 3 or 4 days, so 72 or 96 hours. The critical issues of these assays are, for example, the pH value of the extract that should be around 6. This is the optimal condition for the seed germination, and it's one of the most critical points of this assay for the process of germination. A second important point is the liquid quantity added in the Petri dish. The paper disc should be moist, but there should not be an excess of liquid because this would lead to, for example, anaerobic condition, and this is not what we want. Also, the selection of the seeds during the process of sowing. So we should choose seeds that are uniform in size and intact. And also the selection of the variety used for your assay is very important. And you should select seeds that represent a fast germination rate and high sensitivity. This is an example for you. So here you can see the germination stages of the 𝗕𝗲𝗹𝗹𝗯𝗲𝗶. This is one of the variety we use for the assay. This is the time 0. And this is what we see after 3 days. So after 72 hours. So you can see that almost all the seeds are germinated. And you can consider a seed germinated if it has a root length bigger than 1 millimeter. The number of germinated seeds is noted every day along the whole process. And the length of the hypocotyls and the roots is taken after the 3rd or 4th day of treatment, depending on the variety you selected. This is a second example. It's a variety of 𝗕𝗁𝗁𝗲𝗹𝗯𝗲𝗶. And this is after 3 days, and you can see the elongation of the axis of the seedling. And a third example, it's another variety of 𝗕ข์𝗁𝗁𝗁𝗲𝗹𝗯𝗲𝗶, and this is the time 0. And this is after what we can see after 3 days. So this is an example of how the collected data are represented in a bar chart. And in this study that we published on food and chemical toxicology, we evaluated the potential phytotoxic effects of 4 different hemp varieties water extracts at scalar concentrations. So you can find the concentrations on the horizontal axis. On the vertical axis, you can find the length in centimeter of the seedling measured after the 72 hours. What we see is that remarkable effects in terms of seedling growth slowdown have been obtained with a variety C, but just and only at the highest concentration. So all the other diluted samples, in them we did not observe any significant effect on seed growth, and suggesting preliminary index of extract biocompatibility for the following pharmacological assessments. So let's now move on to the second independent eco-toxicological assay that we perform, the brine shrimp lethality assay. As I already told you at the beginning of this session, this assay is to investigate the biocompatibility or toxicity limits of an extract. So the scientific name of the brine shrimp is Artemia salina. Artemia salina are aquatic microcrustaceans that live in salty lakes and ponds. They are very resistant, actually. And here you can see the development stages of Artemia salina. These are the cysts. These are now the newborns, the larvae. And here you can see an adult specimen. Fun fact, brine shrimps are also called sea monkeys because they have a tail that looks like a monkey's tail. Here you can see the newborns in a water drop. You can see some unhatched eggs on the bottom, and the nauple swimming all around, attracted from light because they supposed to find food where light is. On the right, you can see them under the microscope, and they are very lively boys, we can say. The brine shrimp lethality assay consists in the evaluation of toxicity towards this newly hatched nauple you have seen. This test was previously used in the analysis of pesticide residues, mycotoxins, stream pollutants, toxicant in marine environments, but today it is recognized as an alternative toxicological model for predicting toxicity limits in eukaryotic cells and the assessment of bioactivity of plant extracts. So the first step of the test is the hatching. So brine shrimp eggs are transferred in a vessel with artificial sea water that we prepare in situ with sea salt at a concentration of 25 grams per liter. And they are maintained at room temperature for 24-36 hours under strong light. After this time, we get the active nauple free from eggshells, ready to be collected. Once we have the nauple, we have to transfer gently 10 nauple in each glass tube, and where we can find 5 ml of extract at a scalar concentration in artificial sea water. We run each concentration in duplicate, so we have 2 tubes per concentration. And after 24 hours of incubation under the light, we count the living nauple. and we compare them to an untreated control group. The 𝗕 are considered living if they exhibit light activating movements during 10 seconds of observation. Results are then expressed as a percentage of lethality. And if you plot the percentage of dead shrimps against the logarithm of the sample concentration, you obtain the median lethal concentration, the 𝗕𝗵50 value of the test samples. An extract is considered biocompatible if it shows a 𝗕𝗵50 value greater than 1 milligram per milliliter. In this study, you can see here in these 4 graphs, the same 4 aqueous hemp variety extracts that we have seen in the allelopathy assay before were tested on brine shrimp in a concentration range between 0.1 and 20 milligrams per milliliter. The hemp extracts did not reveal any toxicity in the tested concentration range with 𝗕𝗵50 values all in the range between 1.156 milligrams per milliliter and 2.696 milligrams per milliliter. The second important thing that we get from this kind of study is that the resulting 𝗕𝗵50 value that we obtain gives us an indication of the extract concentration to use for the following in vitro studies on cell lines. And this concentration has to be at least 10-fold lower. So let's say we have an extract that has a 𝗕𝗵50 of 2 milligrams per milliliter. This means that we will use a concentration of 200 micrograms per milliliter in the in vitro assays. So the advantages of these tests are that the test is economic, easy and fast, and that small amount of test material are needed. And an important thing is that several studies demonstrated that there is a good correlation between the results we get as 𝗕𝗵50 value obtained with this kind of test and the results of the acute oral toxicity assay mice. So some critical issue of this test, apart from the great number of advantages, several criteria need to be completed in order to achieve a broad applicability of the assay. These include the standardized experimental conditions in the means of temperature, salinity, aeration, light exposure, pH value of the extracts are also important, and the same geographical region of the cysts, the same age of the Artemia naupi at the start of every test, and positive and eventual negative controls are essential part of the assay in order to check the sensitivity of the naupi and the conformity with the standard procedures. So let's move on to the third eco-toxicological assay that we perform in our studies, that is the Daphnia magna heartbeat rate test. Daphnia magna is a freshwater arthropod commonly found in pools and ponds throughout Canada, in the northern and western parts of the United States, for example. It is used because the contractions of its heart are clearly visible through its transparent body. So you can measure the heart rate at room temperature and then investigate whether the heart rate responds to the plant extract exposure or not. So the first question is, where is the heart of the Daphnia? So these are the antennae. These are the thoracic legs. So could this be the heart? Actually, it isn't, because these are the eggs. The heart is here. So this transparent circle is actually the heart of the Daphnia magna. But let me help you with a video to show you better what I mean. So if you can see it, here there is the heart beating. And important thing is that you have to not confuse the beating of the heart and the movement of the antennae eventually and the thoracic legs. So these are the eggs that I showed you, and here you can see how clearly and visible is the heartbeat of the Daphnia magna. So let's go through the experimental setup. First of all, for this assay, you need a lab partner. And then with a pipette, you have to collect enough Daphnia specimens from your Daphnia culture, place them in a separate glass, give them some time to adapt. And once they have adapted, you have to collect one Daphnia with a pipette and place it on the glass slide and observe it at the microscope. Once you see your Daphnia under the microscope, you have to locate your specimen's heart, so here. And you have to make sure that you are able to see the contractions of the heart. One lab partner keeps time with a stopwatch, and the other counts the heartbeats for 15 seconds. Then you have to record the count in the data table, multiply by 4 the value you get to get the beats per minute. Graph finally the obtained data and analyze them versus a water control or other kind of control. This is a study that we published on processes. It is a very recent study about an association of Tanacetum parthenium and Salix alba extracts in an ex vivo experimental model of migraine. In this study, we analyzed the biocompatibility and the cardiotoxicity of the mixture through the Artemia salina assay and the Daphnia magna heartbeat rate assay. For the Artemia salina assay, the mixture was tested in a range, in a concentration range between 1 milligram and 20 milligrams per milliliter. Specifically, the mixture showed a 𝗕𝗵50 value greater than 2 milligrams per milliliter. Considering this result on the brine shrimp assay, the mixture was tested at the concentration of 2 milligrams per milliliter in the Daphnia magna toxicity test to evaluate eventual cardiotoxicity effects in respect to ethanol 10% solution. So the ethanol representing the reference cardiotoxicity stimulus. At the concentration of 2 milligrams per milliliter, the mixture was able to prevent the cardiotoxic effect of the ethanol induced in the heart rate in Daphnia magna. So as a final point, here you can see another example of a study published on molecules where we investigated the biological activity of Prunus mahaleb extracts in a preliminary way through 2 of the eco-toxicological models you just learned about. On the one side, we investigated the potential toxicity on the lettuce seedlings at a concentration range between 0.1 and 20 milligrams per milliliter. After challenging the seed with extract, we did not find any significant alteration on the germination process. However, at the lowest tested concentration, we observed a significant root elongation, greater than 10% compared with untreated control group. All this was considered as a signal of biocompatibility. On the other side, we performed a brine shrimp lethality assay to confirm the biocompatibility of the water extract at the same concentrations. In agreement with the allelopathy assay, the extract were well tolerated by the shrimps with 𝗕𝗵𝗆 value greater than 10 milligrams per milliliter. Well, this brings me to the end of the speech. Let me just run through the key points again. The toxicity of the plants may originate from different contaminants or from plant chemical compounds, as the phenolic acids we saw at the beginning, that are part of the plant. Various assays are used for the research of potential toxicity of herbal extracts based on different biological models, usually in vitro assays and in vivo assays. However, the recent studies employed efforts for alternative biological assays that include also the 3 eco-toxicological models we just saw together. These toxicity tests are considered a useful tool for preliminary assessment of bioactivity and toxicity of medicinal plants, and they are simple, reliable, and convenient, and lend support for the use of medicinal plants in traditional medicine. So that completes my presentation, my session. Let me just thank the organizer for this great opportunity, and a special thanks goes to you for your kind attention.